Hey guys, welcome back to today's video. So we will be talking about muscles today, specifically the structure of skeletal muscle, like all the different labels and diagrams, and how you get your muscles to contract. So what you get from a stimulus, or your brain sends out signals through nerves and motor neurons, and at the end of the day, what happens to make the muscles contract. So today we're specifically going through that. So first thing you need to know is that for skeletal muscles, they are first of all striated. They're long. They are multi-nucleated. Right, striated means that you've got bands running down like along the muscle fiber cell. Right, so this is one cell, right? Skeletal muscle cells are not like the other cells because of how long it is, right? Because normally a cell will be only about this long, right? Compared to skeletal muscle, they go really long. They've got multi-nucleuses. They've got a lot of nucleuses, right? And they're located peripherally, so like near the outer surface, not in the center of the cell. And that's why a lot of people don't consider skeletal muscle cells as normal cells, and then a lot of people call them muscle cell fibers. But in reality, it's still, it's still a cell. So this is what like the nucleus looks like, and you've got these like bands or going down it, and these are called striations, and this is due to the structure of the protein, basically how they're arranged inside the cell. So and then I'll get to that very quickly. So you go from a fiber bundle or a skeletal muscle, same thing. You go down to muscle fiber cell, right? And then from then on, you've got a lot of different things, right? So you've got your myofibrils, you've got your mitochondria, of course, because you need energy to contract your muscles. You've got your T-tubule, you've got your sarcoplasmic reticulum. So there's a lot of things, right? So I'm just listing the main things that we will cover today as well. Sarcoplasmic reticulum, sarcolemma, right? That's just in the sarcoplasm as well. Okay, so sarcoplasm is basically the cytoplasm of a skeletal muscle cell. Sarcoplasmic reticulum is basically the reticulum in your normal cell, right? So you've got your endoplasmic reticulum. So here we call them sarcoplastic. You've got your myofibrils, which is the muscle, like the actual functional unit, or the sarcomeres, right? So each one in the cell, right? In each one of the cell, you've got these. So you go from these big bundles over here, right, where you've got little like fiber, and then if you go from that, you've got this more singular muscle fiber cell. You've also got these little bundles arrangement, right? And then you go to these, and these are your sarcomeres. So based on how the proteins actin and myosin's arranged, you can see that they kind of form bands, right? This is what gives the skeletal muscle fiber cells this striated look, right? And then we'll get to that right now. So a sarcomere, right, sarcomere is a functional unit of the skeletal muscle fiber, right? It is, so the arrangement of the sarcomere makes it, gives the skeletal muscle the striated look, right? And this is what it looks like. So you've got these middle here, right? These thick guy. And then you've got these thin guys over here. Alright, so this is what it actually looks like. Right, so this right here, this thick part is called the myosin. Right, this myosin filament. Myosin filaments. The thin part is called the actin filament. Actin filaments, right? Actin for thin, myosin for thick. The imaginary line running down the middle is called the M line, right? And then you've got the H band, which is here. H band is composed of myosin only. So just after this actin bit here and just before this actin bit here, if you want to include a bigger one, so it goes from like here, which includes the entire length of the myosin, which have a little bit of actin on the side and actin on the side as well. This guy is called the A band, right? And the I band is just the rest of the sarcomere. I band is just the rest of the myofiber, yeah. I band is the rest, basically, of the actin, so excluding the myosin of the, I'll try to draw it like this. So the I band is this, the A band is myosin, a little bit of actin on both sides, and H band is without myosin, right? So I told you about the imaginary M line running down the middle. Here you've got the Z line. It's really easy to identify because it looks like the Z, right? This is the Z line. So as far as this is all you need to know for labels for A2 biology, right? This is for labels for muscles. Now, you need to know simple facts about skeletal muscles as well. Like they are, so as I said earlier, here they're striated, multinucleated, they're long. Also, they are attached to tendons, right? So, you know, normally how you move around your body, right? If you swing your arm, it's due to the contraction of skeletal muscles. Skeletal muscles are attached to your bones or your skeleton through tendons, right? So, for example, I've got my bone here, I've got my tendons here, and this is your muscle. Right? This is your skeletal muscle. You've got your tendon. This is your bone, and if the muscle contracts, you move your arm up and down or something like that, you know, like a bunch of different movements, right? And basically, normally, you know, for a good example of skeletal muscle is your tricep and bicep, right? So they act in an antagonistic way. So one acts as the agonist and one acts as the antagonist. So if you have your arm, for example, right here, this is your bone. This is your humerus. This is your ulna. This is your radius. Right. Right. This is your humerus. Your bicep is located here. Bicep. And this here is your tricep. Right. And as you curl up your arm, right, so these guys work in different, like, opposite direction, right? So as you curl up your arm, your bicep is contracting and your tricep is relaxing. As you're relaxing down your arm, your tricep is contracting and your bicep is relaxing. So one acts in an agonist way and the other acts in an antagonist way. Okay, so that's how the pair of muscles in your arm work. Now we'll talk about the sliding filament model of the muscles. Right, so what happens is that actually when you pull things, right, so this is your, imagine this is your normal. Like, I'm just gonna draw one single. Got this, right? Imagine this is your normal. Just relax. This is what it looks like when it's contracted. So as you guys can see here, you've got a big H band, right? Here you've got virtually no H band at all, or maybe like a little bit of an H band, because the actin filaments on both sides of the myosin has been pulled together. Right, so this is the sliding filament model contract. So what happens is that, imagine you've got a myosin head, right? So in myosin, you've got a bunch of different heads like this. I imagine this is a big molecule of myosin just going along like that, right? You've got a bunch of heads that can grab onto the actin filament. Right, the actin filaments up here, they grab on by attaching themselves to the binding sites. The myosin, we call them the myosin binding site. So we call these the myosin binding site on the actin for the myosin head to bind to, and it will contract. It will pull backwards, and therefore pulling the actin filament along this way. And this happens to every single one of the actin filament on both sides. So overall, you've got a shorter sarcomere, and therefore your muscles have contracted as a whole, right? And so this is quite a complicated process because if you've got ATP involved as well, and you've also got calcium ions, like a bunch of different things. So we're gonna cover that now, right? So just imagine, right? I'll be revising the cholinergic synapse with you, okay? So this is what happens. Okay. Right. And then imagine it like this, okay? This is what happens. So the first thing happening is that your basically your action potential reaches the end of the presynaptic neuron. And as it reaches the end, it activates the voltage-gated calcium ion channels to flow in. Right? So calcium ions will flow in, right? This acts as a secondary messenger to activate vesicles containing ACH molecules. So these are neurotransmitters, ACH, or acetylcholine, right? This is your neurotransmitter. This is the second thing. So the third step is that they move towards the synaptic membrane of the presynaptic neuron, fuse with it. Yeah, so exocytosis is happening. It will deposit the ACH molecules out into the synapse. They will travel across by diffusion towards the postsynaptic neuron. And the fourth step is that you've got these sodium. So the ACH will bind to receptors on normal sodium channels, right? So these are not voltage gated. When they bind, the sodium channels will open, sodium will rush in, right? And therefore a new, basically a new action potential is generated in the postsynaptic neuron as the membrane depolarizes back to up from negative 70 millivolt, right? So that's how communication, I guess, occurs. That's how action potential is passed on from one neuron to another. And we use the same concept in muscles, right? So this is so as your brain thinks of what you're doing, right? So if your brain is thinking of moving your arm up like this, or moving your arm down, it's sending signals to your motor neuron, and your motor neuron will carry on until it reaches the sarcolemma. So I'll just draw exactly what happens right now for you guys to kind of visualize what happens. So imagine I've got these here, right? So the exact same thing again. Right, so calcium ions will rush in. You've got these vesicle, activate them over here, but instead of a postsynaptic neuron, you've got a surface membrane, and this is the sarcolemma. Right, so this is the surface membrane of your muscle fiber cell, which is called the sarcolemma. And underneath it, you can find different things, right? You can find the mitochondria. You can find one of many nuclei, so one of the nucleus, right? And then you can find, basically, this is your myofibril, which contain functional units called sarcomere. Right, so one of this guy is called a sarcomere, and again, it's composed of actin and myosin, right? So what happens is that as the ACH binds the basically receptor on the sarcolemma, sodium ions will rush in, right? Same thing again, and the action potential will travel across the sarcolemma as well as down T tubule. So I'll show you what it looks like. So this is just a broad overview. Deeper, you've got these things, these structure. Right, these, oops, sorry, the drawing's quite bad. It looks like a cat, I know. Right, so imagine these. This is your sarcolemma. Action potential will travel across like this, travel down T tubule. So this right here is your T tubule, right? And then this right here is your sarcoplasmic reticulum. So these little bubbles here are actually your sarcoplasm. So it's the space, it's the cytoplasm. This entire structure here, if you can visualize it, is the sarcoplasmic reticulum. Right. So as the action potential reaches the sarcoplasmic reticulum, the sarcoplasmic reticulum will release Ca2 ions into the sarcoplasm, and these Ca2 ions will basically activate your muscles. So what happens is, I told you guys that on actin, you've got these little myosin binding site. Remember I told you that, where myosin can bind to. Now, it's not as simple as this, because you've got these little proteins over here called tropomyosin. Right, so these guys basically, like, I'd say wrap around. So all of this guy wraps at the back right here. They go up, they go over, kind of like that. Damn, it's hard to visualize, I know. Go down. So they kind of wrap itself around the actin, the tropomyosin, and covers up the entire, all of the myosin binding site. So I'll draw them again. Binding site. Right, so your myosin can't bind to your myosin binding site on the actin to pull. So therefore, you need the troponin. This is where the troponin protein comes into play. Right, so these protein here called troponin. Right, troponin. What happens is, as Ca2 plus ions is released from the sarcoplasmic reticulum, the Ca2 plus ion will bind to the troponin, and activating a conformational change, resulting in the troponin moving in such a way that as it moves, it pulls away the tropomyosin that has been wrapping all around the binding site, and therefore expose all of those binding sites for the myosin head to snap and bind on, right? And as soon as that happens, a cross bridge is formed between the myosin, the thick filament, and the actin, the thin filament, right? And the myosin will perform what you call a power stroke. So it will pull the actin backwards. So multiple myosin head, right, according to this picture over here, will pull at the same time backwards, right? And basically, pulling the actin filament. And once it pulls backwards, you need ATP. This is when ATP produced by mitochondria comes in. ATP will hydrolyze to form ADP and inorganic phosphate, right? And as ATP hydrolyzes, it provides the energy for the myosin to detach from the myosin binding site on actin, right? And the actin filament. So as you know, so you've got this is your normal. It will power stroke, so kind of bend backwards like this. Then ATP will hydrolyze ATP to ADP plus PI, so that the myosin head will detach to be able to then bind to the myosin binding site further down along the actin to keep pulling, right? So this is what happens when you exercise a lot of ATP is being produced. And when you've run out of energy, right, when you stop exercising, your mitochondria won't produce as much ATP as before. So your myosin binding site basically won't detach from the actin, right? This is what rigor mortis is like. So after you've died. No mitochondria is there to produce ATP, so your muscles are all tensed up. And this is what happens. So that is all you need to know for specifically regarding skeletal muscles. Contraction, or the different labels and stuff, the models. And that is basically it for A2 Bio skeletal muscle fibers. Thank you for watching today's video, and I'll see you guys in the next one.